Good afternoon and welcome to the ATP-Bio public webinar series. My name is Ellie Gardner, the Communications Manager for ATP-Bio based at the University of Minnesota here in Minneapolis. Just a couple of things before we get started. Today's webinar, as well as all our past webinars, will be posted to our YouTube channel. We'd like to ask everyone to please share their video, but keep your microphones muted throughout the presentations. And then lastly, I'd like to invite you to please save the date for our next webinar on May 5th, featuring Dr. Matt Palpalm. And it's not advancing. But in any case, so May 5th, save that date. And without further ado, I'd like to hand it over to our moderator for today. Desiree, the floor is yours.
Hi, everybody, and welcome to the ATP-Bio public webinar series where 3 trainees will be highlighting their innovations in endothelial protection and ice-free preservation across blood and organ systems. My name is Desiree Gutierrez, and I will be your moderator for today. I'm a material science and engineering student at Texas A&M University, the newly elected co-chair of the ATP-Bio Student Leadership Council, and an undergraduate researcher in Dr. Guillermo Aguilar's lab working on laser nanowarming for cryopreservation applications. During these presentations, feel free to jot down any questions you may have. We'll have a Q&A session after all 3 speakers are finished. Our first presenter today will be Abhilash Ojha. He's a PhD candidate at the University of Minnesota working in Dr. Chris Hogan's lab. His work focuses on spray cooling thermomanagement, nanoparticle modeling, and flow visualization. Today, he'll be presenting his work on aerosol-based cryopreservation. I'll hand it off to you, Abhilash.
Yep. Thank you, Desiree, for the introduction. And is my screen visible, the slides? Yeah, okay. So, yeah, I would be talking about aerosol-based cryopreservation. So we know that cryopreservation is a process of cooling the biological sample below its glass transition temperature rapidly. And this rapid cooling rate is called critical cooling rate. And when the sample is cooled above this critical cooling rate, so it achieves a temperature state called vitrified state. And this vitrified state, in this vitrified state, it can be stored and preserved for a long period of time in extended storage. So to cryopreserve the samples, biological samples, we need a very high cooling rate. And sometimes this cooling rate can be as high as 10 power 6 Kelvin per second. Like for water, for example, for water, it's 10 power 6 Kelvin per second. And it may not be feasible to achieve this cooling rate. So we add cryoprotectants to the samples, and these CPAs, they bring down the critical cooling rate so that it's possible and feasible to cool them at those rates. But these CPAs, at the same time, they are toxic as well. And once we use the CPA and cool them above the critical cooling rate, we can vitrify them. This is how the vitrified state looks like. But the problem with this method and the methods being used in the literature is that they have a limited throughput. Like here, if we plot the cooling rate achieved in the literature with the volumetric throughput on the x-axis and cooling rate on the y-axis, we see and we show all the literature data points on this plot. So on x-axis, on the extreme left, we have a low throughput but very high cooling rate. But on the extreme right x-axis, we have high throughput but low cooling rate. But for these technologies to be clinically translated, we need to be in a region of high cooling rate and high throughput, like this one. So we developed a technology that utilizes aerosols and to achieve the clinical translation. And aerosols are two-phase system that comprise of solid or liquid particles suspended in gas. And since they are small particles, droplets, and they behave as a lump system, so their cooling rate is inversely proportional to the radius. So if we have small droplets, we can cool them faster. So if we cool the aerosol droplet faster, so we would need less CPA to cryopreserve. And since aerosol technology is based on atomization, so it is highly scalable. So we can get the high throughput as well. Based on our experiments, when we plotted our experimental data point on this literature plot, we see that we are in the clinically relevant cooling plus throughput regime, as shown by the green asterisk points. So the experimental setup that we used for this process is shown here. So at the top, we have two syringe pump, which pump the CPA and the cells, and both of the solution, they meet in the single tubing, and the cells are loaded with the CPA just before being sprayed, like shown here. And then we have a aerosol generator. So when the syringe pumps the cell solution, it is forced through a orifice, which is shown by the red circular disc here. And when the cell suspension passes through the orifice, it forms a liquid stream. And this orifice also is vibrating. So this vibration of orifice perturbs this liquid stream and it generates the droplets, like shown here. It generates monodispersed aerosol droplets. And these monodispersed aerosol droplets are forced to go into the stream of liquid nitrogen, which is coming out of the side nozzles as shown here. And this liquid nitrogen is at the boiling temperature. So the aerosol droplets, they collide with the liquid nitrogen droplets and they pre-cool. And they hit a plate as shown here, which is a copper plate. And as soon as they hit the plate, they are vitrified and then collected. And then they can be used, they can be stored, and then they can be later rewarmed and can be used. So in order to quantify this process, so we image the aerosol droplets like shown here. On the left-hand side, we have aerosol droplets from 10% weight by volume ethanol using 100 micrometer orifice. So here we see that these droplets, they appear as white color. It means that ice has completely formed in them. And here the cooling rate now is lesser than critical cooling rate. That's why they appear frozen. But on right-hand side, we have another set of experiment with 13.75% ethanol weight by volume and 100 micrometer orifice, where we see that some of the droplets are appearing as white and some of the droplets are transparent. So the droplets which are transparent, they are the vitrified droplets. So because we can see that some of the droplets have started to vitrify, we can say that the cooling rate for this concentration is in the ballpark of the critical cooling rates. Now from these two images, we can extract some data that can help us to quantify the critical cooling rate. We can get the size distribution. We can get the state of the droplet, whether it is vitrified or frozen. And for that, we used a image segmentation model, machine learning model. The reason for using the machine learning model was because the intensity of these droplets, it changes from experiment to experiment. And we have the experiment with different size droplets. So the model can learn about the features of the droplet and it can find the size. And it would be independent of the intensity of the images as well. And we can automate it to process thousands of images. So the way this model works is it takes a raw image as shown on the left-hand side, and it processes it and it gives the edge of the droplet because it has been trained to give the edges. And simultaneously, it has another channel which gives the centroid of the droplet also. The reason for extracting out the centroid is to differentiate between two overlapping droplets. And after that, based on the intensity of the droplets, it can classify the state, whether the droplet is frozen. If the intensity is high, it is classified as frozen. If the intensity is low, it is classified as vitrified, as shown here in green and pink color. The pink color droplets are vitrified and the green are frozen. And since we already have the centroid and the edges, we can find the size distribution. And we came up with this mathematical formulation that to quantify what is the fraction of droplet that is vitrified in that image. So it takes the radius of the vitrified droplet and then cubes it and divide by the radius of the vitrified droplet plus the frozen droplet. So using this formulation, we can find that in a particular image, what's the total fraction of vitrified and what's the total fraction of frozen droplets. And using this equation, when we plot it for different size of orifices, we get a variation like this as shown on the left-hand side. And here I'm showing the experiment with two different orifice sizes, 50 micrometers and 100 micrometers. And since 50 micrometer droplet is smaller, it will cool faster also. So on the y-axis here, I have crystallized fraction, meaning it's 1 minus the vitrified fraction that I have shown on the previous slide. So if this value is 1, it means that there is 100% ice formation. If this value is near 0, it means that there is less ice formation, it's mostly vitrified. So we can see from here that 50 micrometer orifice vitrifies faster than 100 micrometer orifice. And if we take this curve and differentiate it, we can find the cooling rate experienced by this droplet, as shown on the right-hand side figure. And from here, we can see that the cooling rate experienced by the 50 micrometer droplet is about an order of magnitude higher. 100 micrometer droplet experiences about 0.9 into 10 power 5 Kelvin per minute, but the 50 micrometer droplet is experiencing about 1.2 into 10 power 5 Kelvin per minute. Also, we also studied the effect of the flow rate because we have a nitrogen aerosol coming into which the droplets are injected. And the rate at which this nitrogen aerosol is coming out, the nitrogen flow rate, will also affect the cooling rate experienced by the droplet. If we have higher flow rate, like on this images, I have shown 3 images from low flow rate to high flow rate from left to right. So if we have higher flow rate, there are more nitrogen droplets coming, and there would be more collision happening between the droplets and the nitrogen droplets. And the nitrogen droplet will boil on the surface of the cell droplets. So if we plot the cooling rate distribution for these 3 flow rates, we can see that the higher flow rate gives the highest cooling rate, about 2.5 into 10 power 5 Kelvin per minute, but the lower flow rate gives about 1 into 10 power 5 Kelvin per minute cooling rate. And we, to give a proof of concept of this technology, we tried cryopreserving red blood cells and compare it with the literature. So we took the RBCs and then we loaded them with 2.5 molar glycerol, because in the literature also the hemolysis of RBCs at 2.5 molar glycerol has been reported, so it's easier for the comparison. So we loaded them with 2.5 molar and 4 molar glycerol, and then we sprayed them using 200 micrometer orifice, and then we rewarmed them back into the same CPA solution as they were loaded in. And we can see the comparison that our work gave the hemolysis of 12%, while the literature work that we had reported in Cryobiology 2024, it gave the hemolysis of 10%. So this proves that there is no difference between spraying in terms of hemolysis. There is no difference between spraying and the methods used in literature. But this work that we have, it can achieve higher throughput. We can process like 3.8 millimeters per minute of RBCs, or sometimes we can go higher as well. So summarizing everything, we saw that using aerosols, we can achieve the clinically relevant cooling plus throughput regime. We can have higher cooling rate as well as high throughput as well. And we see also that the smaller droplets, they vitrify faster. The orange color curve is for the 50 micrometer droplet. It vitrifies faster because it's smaller. And we also have shown the proof of concept for this technology with RBC droplets. So yeah, that's it. Thank you. Any questions?
Thank you, Abhilash, and we'll go ahead and take those questions at the end. So our second presenter is Luke Boudreau. Luke completed his undergraduate studies at the University of Massachusetts Amherst, earning degrees in both chemical engineering and biochemistry. After graduating, he worked as a research technician at Massachusetts General Hospital in the molecular and cellular bioengineering group under the guidance of Dr. Berk Usta. He'll be presenting on the optimization, benefits, and scale-up of red blood cell supercooling. Take it away, Luke.
There I am. I realized I had to unmute. All right, one second. Let me just pull up my laser pointer and hopefully everything else will go off without a hitch. Everyone can see my screen all right? Perfect. Thumbs up. Thanks, Brad. Hi everyone. Good morning, good afternoon, good evening. My name is Luke Boudreau. As Desiree said, I will be presenting on red blood cell supercooling, optimization, benefits, and scale up, work I've been involved with over the last 3 years. Um, go. Before I begin my talk, I always like to give a shout out to the man himself, Dr. Charles Drew, who began the concept of blood banking during World War II, I believe, and allowed us to take advantage of the one of the few regenerable biological um, transfusions we have. And he has essentially allowed us to use RBC transfusion continuously. It's the most common cell therapy, and it helps us circulate oxygen throughout our body continuously to prevent ischemia throughout our body, and is used consistently. Over 120 million units are donated every year. However, this need continuously fluctuates, especially with disaster, and global demand is rarely fulfilled. The current problem being that the most basic form of storage, cold storage, at 4C is mandated to only be used. The units need to be used within 6 weeks of donation, excuse me, or 42 days, and only hemolysis is used as a parameter for sample quality. Once again, let me reiterate, the modern approaches, as we know right now, are cold storage in the low above zero range from about 1 degree C to 6 degrees C that allows us to have a high yield of viable cells for that limited storage time as I manage. However, the trade-off is we're able to use these very easily. You can take a bag right out of the fridge and put it straight into a patient as necessary. Alternatively, the other method is cryopreservation. Similar to how Abhilash presented, adding CPAs to your substance, freezing it down to -80 and freezing it. However, this is only generally used for rare cell types or for long-term storage of blood cells. And, it is a slow and, there's a lot of processing required to actually use the blood for transfusion following unfreezing. And issues with this were seen greatly during the September 11th attacks when there was enough blood, but not enough ready for use. Our goal with supercooling has been to create a method of storing blood for longer periods than 42 days without a decrease in yield and quality from donation for practical use by storing in the high sub-zero temperature regime, namely from 0 to -5C. And you might be asking me, Luke, how would you do that? Because below 0, blood should be freezing like any other substance that has water. And the answer is through a rather neat process that my lab developed over time, known as mineral oil surface sealing. And by adding a layer of mineral oil on top of our red blood cell suspension, we're able to stop ice formation at the air-water interface by decreasing contact angle between air and water. And as a result, we're able to store red blood cells for the majority of the time, 1 milliliter samples with a layer of oil at various temperatures without ice formation. In our case, we generally are using +4 cold storage to see the standard process, -4C, which is where we will generally, or our optimum storage temperature, and -8C if we wish to exacerbate cold storage injury. And after our storage time points, we can analyze for multiple different parameters, namely hemolysis, as I mentioned before, but also red blood cell indices, or the size and hemoglobin of red blood cells, as well as metabolism or untargeted metabolomics and lipidomics, as we've done previously, as well as other assays following self-fractionation. Oop. And so to begin, our solution, so to speak, became a dual-pronged effect. A, using new additive solutions, and subsequently supercooling as I described. Our initial paper described how was a comparison between many additive solutions in literature, ESOL5, AS7, CPDA1, and SAGM, which is CPDA1 and SAGM being most commonly used. Over 6 and 10 weeks, we actually saw that at +4 and -4, ESOL5 was the most beneficial solution for storage. ESOL5 being a more newer generation solution that it was alkaline instead of acidic, chloride-free instead of using chlorides or sodium salts, and buffered rather than non-buffered. And as a result of this initial set of experiments, we chose ESOL5 as a baseline for optimization going forward, namely to be add antioxidants as was in our initial publication, and we have since explored others such as other substances such as amino acids. However, the great goal of our supercooling is to depress metabolism and at some point beat cold storage. And as you can see, we initially identified a crossover point at some time point between what is likely 16 and 18 weeks. And our hope is that by slowing metabolism, we can, and optimizing our solution in the future, we will be able to lower hemolysis to beat cold storage over time. And although we were not able to fully decrease hemolysis down to usable levels with our initial set of experiments, we initially asked what exactly is going on with our red blood cells. So we were able to complete both metabolomics and lipidomic experiments with collaborators at the University of Colorado Anschutz, namely with Dr. Angelo D'Alessandro, where we wanted to both elucidate an injury mechanism as a result of our cold storage and highlight possibly novel benefits as a result of our lower temperature storage along with our novel additive solution. And so we found a couple of things. So we compared our worst performing solution, as I showed previously, SAGM, to ESOL, our best performing additive solution. And while we saw the same trends in hemolysis, one novel benefit we saw was a incredible conservation of 2,3-biphosphoglycerate, or 2,3-BPG, the molecule that allows for oxygen to be released from red blood cells and gives it its purpose, as well as a conserved energy charge or an amount of ATP adenylates within the substance. Namely, this 2,3-BPG conservation has also led us to collaborate with ATP-Bio industry sponsor Biochip Labs to do functional assays on our red blood cells going forward. That work is currently ongoing. In terms of our RBC lipidomics, we were actually able to see injury mechanism, namely membrane instability, and specifically loss of phospholipids and polyunsaturated fatty acids over the course of storage time. However, we also were able to find a solution for this, that being supplementation of small amounts of ethanol volume by volume. In this case, it was 1, 2, and 4% supplementation of ethanol into the additive solution, not into the full solution, where over time, over the course of storage, we were able to conserve this decrease of, as shown here, lysophospholipids over the course of storage. Subsequently, we also wanted to see the effect that inclusion of ethanol had on alcohols, or on hemolysis. And what we saw that increasing alcohol in the additive solution also subsequently decreased hemolysis in a dose-dependent manner up to 20%. We chose not to go beyond 20% because really anything more than 8% is going to be toxic anyways for transfusion. And as I said earlier, we wanted to create a system that would be easily transfusible following storage. Subsequently, we also asked, could it go longer than our 42-day time point that we generally store at? And the answer is yes. sorry, supplementation of 4, 6, and 8% ethanol in this case, as we showed, conserves the effect of decreased hemolysis up to 11 weeks of storage. And so we ask ourselves, now that we have a solution for decreasing hemolysis and ensure it over the course of storage, can we scale? And so we then contacted our friends at Instant Systems, namely Ojha, who is also an industry sponsor of ATP-Bio, and we started with our 1 milliliter samples of red blood cells, scaled from 1 milliliter to 15 milliliters to 30 milliliters to 50 milliliters, and finally to 100 milliliters of packed red blood cells in supercooling. And we were able to store these samples to their regular 42-day time point, aka 6 weeks, and then subsequently to 100 days without ice formation, without oil, and we saw subsequently in a decreased hemolysis as we increased in volume. And we hypothesized that as a result of more contact with our additive solution, as these were stored horizontally in our coolers. Additionally, we use 6% ethanol as a baseline for storage here. We saw that in combination with increasing our volume to 30 milliliters, and for reference, each one of these is a different donor. We saw combining the ethanol effect with the decrease in hemolysis that we saw, we are able to store just at hemolysis levels that are equivalent to or just as good as our +4 controls. So in summary, we were able to create a platform for supercooling screening and optimization in the 1 milliliter samples. Subsequently, we quantified unique metabolic character and have begun to understand mechanisms of injury in our supercooled samples. And we have subsequently formulated and characterized our additive solution to decrease hemolysis and reduce membrane injury in the short and longer term. And as a result of all this, scaled the system of supercooling to larger clinical volumes for possible use in the clinic at some point in the future. With that, I want to thank all of my collaborators, specifically Matilda Holtz, who is my partner in crime, who may not be in the call, as well as everyone else, especially our Canadian collaborators, Jason Acker and Shaka William, and all previous and current members of the team. Mesh, I know you're here, so I'll also thank Mesh because he's here as well. So that's my talk. Oh, I lost the question slide on the way there, but we're doing questions at the end. So thank you very much, Desiree. I will hand the floor back to you. Thank you, everyone, for giving me the opportunity.
Thank you, Luke. And go ahead and feel free to type your questions in the chat box. We will be going over them at the very end. Our final presenter is Brad Ellis. Brad earned his undergraduate degree in bioengineering from the University of Pittsburgh and his master's and PhD in the University of Notre Dame. He is currently a postdoctoral fellow at Massachusetts General Hospital under the guidance of Dr. Bashak Oygun, studying preservation injury in endothelial cells. He'll be presenting on reducing endothelial preservation injury through oxidative stress and apoptosis mitigation. Over to you, Brad.
Thank you, Desiree. Just double checking as everyone else has. Everyone can see my slides, correct? Yes. Okay, cool. Awesome. All right, thank you. Yes, so thank you so much. As mentioned, I'm Brad Ellis. I'm a postdoc here at MGH under the advisement of Dr. Bashak Oygun. And thank you very much for the opportunity today to present my work on reducing endothelial preservation injury through oxidative stress and apoptosis mitigation. So how this project started was looking at the problem of a lack of livers for transplants. So with this audience, probably don't need to go too much in depth with this, but as most of us know, the wait list for all organs, but in this instance here, specifically liver, is significantly increases every year. And even with this increase, with the supply far outstripping the demand, approximately 10% of livers that are recovered for transplant actually don't end up going to or end up not being transplanted. And a lot of these livers are actually what are known as extended criteria donors, or individuals who have donation after circulatory death, had fatty livers, or the livers were older in age. And even with novel preservation systems, such as extended preservation as well as machine perfusion, a lot of these marginal livers are actually still undergoing significant preservation injury, specifically through ischemic reperfusion injury, to where these new methods are not necessarily capable of making these marginal livers transplantable. So with that, a lot of work has been done looking at the liver as a whole organ, as well as hepatocytes, the main cell type of the liver. However, there's been little work looking at the non-parenchymal cells, or NPCs, of the liver. These are the other cells of the liver, which include the Kupffer cells, which are liver macrophages, our immune cells, hepatic stellate cells, which are involved in fibrosis and scar tissue formation. And then most importantly for our lab and for the work shown here, the liver sinusoidal endothelial cells, which are highly specialized microvascular endothelial cells that line the liver capillaries. So these are the second most popular cell type in the liver. And like mentioned, they line the liver capillaries. So anything that the liver sees, the LSECs are essentially going to be seen first. That goes for cryoprotective agents, lack of oxygen due to reduced blood flow, etc. One other thing I should mention that will come into play a little bit later in the presentation is that the LSECs themselves are fenestrated, meaning that they are quote unquote holy, and that with that, large proteins can actually pass through passively from the liver into the from both ways through the liver into the capillaries and vice versa. So with this led us to looking at trying to figure out, how to evaluate the impact of static cold storage, which is the current standard preservation method for livers, or SCS, mediated preservation injury on LSECs, and compare that to other hepatic cells and investigate potential therapeutic strategies to mitigate this injury. So how we did that was we did this on essentially a cell-by-cell basis. So what we did was we took rat livers and isolated the 4 cells that I mentioned earlier, the hepatocytes, the LSECs, the stellates, and the Kupffer, and then cultured them individually to basically see what these cells did on an individual level without any crosstalk between them. We then put these cells in under essentially a transplant model where we stored them in static cold storage for 1 or 3 days, and then gave them and then used them as quote unquote transplant to mimic the ischemia reperfusion injury. And then following the recovery under standard culture conditions is when we did our viability and functionality measurements. So the first thing that we noticed was when we looked at these cells is we saw that LSECs had a different response to the other hepatic cells. So if you look here in the blue bars being LSECs, we saw, especially when we extended the storage length time to 3 days, we saw that the LSECs had a greater survivability or higher viability than all the other hepatic cells, hepatocytes, stellates, and Kupffer cells. We saw that this trend continued both for metabolic activity as well as ATP levels. Additionally, when we looked at programmed cell death or apoptosis marker, caspase, we saw that the LSECs had significantly lower levels than other cell types. So with this, we were able to see that the cells are surviving under preservation injury better. However, we still saw significant reactive oxygen species, which is a hallmark of ischemia reperfusion injury, or in this case, specifically oxidative stress. So we were wondering that, yes, these cells may be surviving, but how well are they functioning? So with that, we looked at LSEC-specific assays, including their scavenging ability. We looked at this through acetylated LDL uptake, which is more general endothelial cell scavenging, whereas LSECs are one specific function of LSECs is that they will scavenge for heparin. So then we looked at that as a more LSEC-specific functionality. And we saw that when we had 1 day of preservation injury, we saw a significant increase in both scavenging abilities, whereas when we increased the preservation time to 3 days, we saw that only heparin attachment levels were increased, whereas acetylated LDL uptake fell back off down to fresh levels. We also looked at how well these cells were able to maintain their membrane. So as I mentioned earlier, the LSECs themselves have fenestration. So when we culture these cells, they already have some inherent permeability. However, when we introduce preservation injury to these cells, we see that even at 1 day preservation injury, we see a significant increase in membrane permeability, showing that the integrity of the monolayer of these cells is damaged either through cells sloughing off or an increase in fenestration, most likely, as I'll show in the next here, from cells dying and sloughing off. So we wanted to look more specifically at how these cells looked following our storage. So using TEM, we looked at the cells morphology under fresh 1 day static cold storage as well as 3 day static cold storage. And we saw that the longer we kept these cells in preservation, the more damage indicators that we would see. And this would include nuclear membrane separation here in red, chromatin aggregation, as well as phagosomal content and increased lipid content. So this showing that even though these cells are surviving, they're undergoing a significant amount of injury. With that, we wanted to look farther into whether or not this injury is leading to increased secretion of chemokines and cytokines, specifically for a common motif in ischemic reperfusion injury. And with this, again, we saw a significant increase in several chemokines and cytokines, including interleukins, some general cytokines, including, interestingly, cell proliferation cytokine EGF, which I'll mention as well later. And then as the big one being an increase in an inflammation response in these cells. So again, further showing that these cells, though surviving, are still undergoing significant damage. With that, we wanted to then look closer into what kind of damage that was. What kind of was this through? What type of cell death was occurring that was hurting these cells? So with this, we looked at the gene expression, and we saw first, following 1 day of static cold storage, we looked at cell death gene expression as well as oxidative stress. And 1 day, specifically looking here at cell death, we only see a downregulation in genes, interestingly. And a lot of these genes are actually associated with autophagy and necrosis. And then with oxidative stress, we see, again, a downregulation in several different genes, specifically through some oxidative stress peroxidases, some oxygen transporters, just basically the whole gamut of oxidative stress. When we increase the storage length to 3 days, though, we see a significant upregulation in apoptosis-related cell death genes. And then again, significant changes in oxidative stress-related genes, indicating that these cells are undergoing significant oxidative stress, which in turn is leading to apoptosis. So with this, we see that preservation injury is mediated through both apoptosis and oxidative stress for these for LSECs. With that, we then asked ourselves the question of, okay, we know that this is through apoptosis and oxidative stress. Can we help LSECs through preservation injury by lowering apoptosis and oxidative stress? So if we go back to our original experimental plan here, what we tried to do here was inhibit apoptosis through the addition of a small molecule anti-apoptotic cocktail called CEPT, which is a combination of several small molecule markers, including chromin-1, imricasin, polyamine supplement, and trans-integrated stress response inhibitor. This has seen success in promoting cryopreservation of induced pluripotent stem cells, as well as our group has published a paper showing that this helped for warm ischemia in whole livers as well. And then to help for oxidative stress, we added in vitamin E analog that is cell permeable called Trolox, which is a very strong antioxidant. And we wanted to see what the combination of these treatments did to help LSECs following preservation injury. And lo and behold, when these treatments actually were able to rescue LSECs from preservation injury, specifically through significantly increasing viability, as well as metabolic activity,
in ATP levels. And this was also coupled with a decrease in apoptosis marker caspase 3,7 activity, as well as a decrease in ROS production. However, everything wasn't perfect. So when we looked at the actual LSEC functionality, the results were not as drastic, but we did still see some beneficial effect, including a decrease in heparin attachment, as well as I mentioned earlier, the decrease in EGF secretion of these cells following treatment. However, when we looked at other markers, we saw no changes in any of our other functional markers that we looked at, including acetylated LDL uptake, or changes in permeability, or changes in chemokine and cytokine secretion. When we looked at the cell morphology through TEM, we did see a decrease in nuclear membrane cell separation, as well as lipid content. However, we did still see some damage indicators, including some lipid content, as well as in this example here, some chromatin aggregation. So overall, the results from the study, we were able to conclude that LSECs have a different response to static cold storage than other hepatic cells, that this response is driven through oxidative stress and apoptosis, and that by inhibiting apoptosis through SEPT and oxidative stress through Trolox, we're able to increase LSEC survivability and to some extent functionality following preservation injury through static cold storage. And these results really highlight the importance of understanding LSEC-specific injury mechanisms, specifically as a way to find novel therapeutic treatments to help the whole organ through preservation injury. I also wanted to mention that this project has kind of branched off as well. So seeing how the LSECs, a highly specialized endothelial cell, responded to preservation injury, and how this was different than other hepatic cells, we were interested in how this works with looking at different endothelial cells, specifically endothelial cells from large blood vessels and small blood vessels. So this project has now spanned out to looking at all different types of endothelial cells. And some interesting results we've seen from that is that endothelial cells from the microvasculature seem to be have much greater trouble dealing with preservation injury. We've also used this as a these treatments to look at a ex vivo model of this through vascular composite allograft transplant through rat hind limb, to see whether or not what we're seeing here in vitro is maintained ex vivo. And then also we're working in a collaboration through Dr. Kevin Healy at UC Berkeley, as well as Dr. Young-Dok Kim at the Korea Institute of Science and Technology, to increase the complexity of our in vitro model to include, basically to make microfluidic devices to include shear stress, and with these microfluidic devices to be able to have on-chip measurement, including membrane integrity, as well as cellular health. So with that, I would like to acknowledge my advisor, Dr. Bashak Uygun, for all of her help, as well as current lab members, Alpern Abachi and Elizabeth County, as well as specifically past lab members, Huyun Chen and Mohammad Reza Mujudi, who have helped a whole bunch with getting this project off the ground, as well as our collaborators, Korka Ugan, Heidi Ye, Albin Longchamp, Martin Yarmush, and Mehmet Toner, as well as the funding sources, the NSF, NIH, and Shriners. And with that, since I am the last presentation, I can give the slides. So thank you very much for your time. And I'm assuming we'd all be happy to take any questions that you do have. So I'll pass the floor back to Desiree. Thank you so much.
Thank you, Brad and Abhilash and Luke, for taking the time to present y'all's interesting work. We'll now have our Q&A session. I'll first go through these questions that we have here in the chat, and afterwards, just feel free to use the raise your hand feature on the Zoom, and I'll go ahead and call on you to answer those questions. So first, for Abhilash, we have from Gamid. How are you ensuring the internalization of CPA in prepared cell suspension prior to being aerosolized? What parameters are you using to decide whether the cells have been incubated long enough in suspension/solution? Balance between toxicity and internalization.
Yep. So I can share a slide for that. So I'm exactly following the methodology that Liu and all used in their paper Cryobiology 2024. So they took the 2.5 molar glycerol and they loaded the cells RBCs dropwise. So I'm using the same, loading them dropwise over 10 minutes of time. And then I'm measuring the hemolysis after that process. So the purple hemolysis that we see, I mean, it's viability, which is 1 minus hemolysis here. So we can see that the viability is 95%. So I'm ensuring that the cells have been properly loaded and incubated with the CPA after getting the hemolysis, calculating the hemolysis. But I'm loading them dropwise to make sure that there is no osmotic stress in them.
For
Yeah.
Abhilash as well, from Adam, can you comment on how you acquired the droplet images? It seems like it would be challenging to ensure the small droplets remain below the glass transition temperature for imaging.
Okay. Yeah. So for the imaging, we are using, so I will go back to my schematic. Yeah, so after we spray the droplets and collect them on the mesh as shown here on this schematic image. So because liquid nitrogen is flowing through them, so they wouldn't de-vitrify. They would be in the vitrified state or the state that they are. And then I'm taking those droplets and then putting them in a liquid nitrogen bath on my imaging setup here. And I'm suspending them in the liquid nitrogen bath while imaging also. So the thing is that they are throughout the process after the spray and when they are imaged, they are always being suspended into the liquid nitrogen. And while imaging also, they are into the LN2 bath. So that's how I'm ensuring that they don't de-vitrify. It's difficult to sometimes image them because of the vapor forming, but after you wait long enough, the vapors move away and then we can image them.
Last one sorry, go ahead.
Yeah, I read that one. So for the question that how much the droplets are being cooled into the before hitting the plate, like when the droplets they come out of the orifice and before hitting the plate, what's how much they are being cooled. So for that, I would say that this liquid nitrogen stream that into which the droplets are being injected, they are at the boiling point of LN2, and there it's LN2 vapor plus LN2 droplets, and the cell-laden droplets, they are colliding with LN2 vapors. So it's a multi-physics phenomena happening. It's like we have an equation that can find it. I don't have a exact fraction how much of them is being cooled, but all I can say that because they are colliding with LN2 droplets, so most of the cooling is happening into the in-flight phase before they hit the copper plate. And we have this mathematical model that can predict what's the cooling rate being experienced in flight. So on the left-hand side of this equation is the droplet mass and the cooling rate dt by dt. And the first term on the right-hand side is the convective cooling term that is coming from the LN2 vapors during the flight. And the second term here is the collision term that's happening with the LN2 droplets. So combination of these two terms gives the cooling rate that is happening in flight. And we have also shown it on the plot of critical cooling rate versus volumetric flow rate. So the blue color region that we see, this is coming from this equation. So we can estimate the cooling rate that's happening in the flight using this equation. But I don't have right now the fraction. So the other way that I can answer this and explain it is most of the cooling is happening in the flight is by this second plot. Here I have shown the cooling rate distribution for two cases. They are being sprayed into the LN2 bath, but the first case is the pre-cooling case, which is the blue one, means that LN2 stream is coming out of the nozzle. And in the gray one, which is no pre-cooling, means that the droplets are directly being injected into the LN2 bath. So we can see that when there is no co-flow present, no pre-cooling LN2 is present, then the cooling rate is less. But when the pre-cooling LN2 is present, then the cooling rate is like more than double. So we can say that most of the cooling is happening in flight. Yeah.
I wasn't I wasn't asking the cooling rate. What's the temperature by time? So you're saying the cooling is done in the gas phase.
Yes.
So why do you need the cold plate to bang the cells to the cold plate? You just cooled it already.
No, so they are not vitrified. They vitrify only when they hit the plate. But
That was my question. So what was your final temperature when it reaches the cold plate?
So it would be near boiling point of LN2, not the LN2 temperature, but near boiling point of LN2. But I don't have an estimate of that.
What's the temperature of it?
It would be,
I'm asking the droplet temperature.
yeah, the droplet temperature itself. It would be boiling point of LN2, which is somewhere around -195
Yeah.
C. Yeah.
So it's already vitrified. Why do we need to cool further than -195
So
to get vitrification? I'm missing the whole point of the process.
Yeah, it's not vitrified. The thing is that if we don't pre-cool it, then when it hits the liquid nitrogen, the plate, so then there would be some vapor forming around it.
I know Leidenford's effect and all those things. I'm not asking those questions. I'm asking a very simple question. You're trying to cool something.
Yeah.
Part of the cooling, I don't care about the cooling rate, okay? Part of the cooling happens in the vapor phase. The rest happens when it touches the cold plate. Is that right?
Yeah.
What's the temperature before, just 1 millisecond before it touches the cold plate?
I don't have a measurement of it, but it would be like less
calculation. You will not have a measurement, I know, but calculation.
Yeah, so but we can estimate it from this equation here.
Yeah.
Abhilash, maybe you take a peek at it and send him a note later.
Yeah.
But I think it's equilibrated, Mehmet.
Equilibrated with the -195.
I think so, yeah.
And why do you need so my question is, why do you need to smash it onto if it's already vitrified, why do you have the second part?
I think that this might be something, a language thing, or maybe I missed it too, but I thought you were using that as a collecting opportunity.
No,
not for cooling. Collecting.
Yeah,
okay.
It shouldn't be for cooling. It should just be for collection.
Okay, that explains it. Yeah, that's why I was curious. I actually thought it was very cool to do two-stage cooling, but then I was saying it's going to equilibrate by the time it reaches there.
Yeah.
Okay. So I got it. Sorry, Abhilash, I just didn't clearly follow your talk.
no worries.
Thank you.
Okay, we can go ahead and move on to Luke. Luke will go ahead and read the questions from the chat, and then afterwards go ahead and ask him any questions y'all have.
For sure. So first from Gurji, do you test different cooling rates for supercooling if they have any impact on stored blood? The simple answer is no. We have not. But we're also generally only changing the temperature. We're changing it very little. In the short term, we have measured hemolysis from like day 0 to day 1 and day 3. And until about day 7, there's not a significant change in hemolysis between like a +4 and a -4. Of course, there's a lot of cold injury at -8. But in the two most important comparisons, we're not seeing any significant differences. Unlikely that it is. We have not tested whether or not cooling rate has affected it. So yeah.
Luke, I believe you have one more from Daniel.
Was that me or is that for Nope, I think that's for Abhilash. Because that has to do with LN2.
Okay, yes. Abhilash, go ahead from Daniel.
Brad, how about you answer your question while Abhilash reads it for a second?
Yeah, sure thing. Sounds good. Okay, so Zhanghu asked for Brad, what are your thoughts on using SEP to help reduce CPA injury? Is the sept only helping reduce the apoptosis? And then will the sept work for organs other than the livers? I'll answer the last part first. So yes, sept has been shown to be helpful in other organs, I believe. So the main thing was it was originally used to help the cryopreservation of human-induced pluripotent stem cells, and helping them actually survive freezing and thawing. Since for anyone that's done any iPSC culture knows that they're the absolute biggest prima donnas when it comes to being cultured. They will die if you look at them wrong. But also they've shown some, if I remember right, there's been some papers that have looked at them in culture for helping cardiomyocytes as well. So yes, so I would believe that STEP would work for other organs other than the livers. However, regarding the CPA injury, so SEPT is specific. The four additives have basically serve very specific functions, being small molecules. So chromin 1 is a ROCK inhibitor, which is going to help more with cell attachment. Imricasin is going to be the caspase inhibitor that really helps with the apoptosis. Polyamine supplement is it's a media supplement that helps with cell growth. And then the integrated stress response helps with basically cells dealing with stress. It inhibits the cell stress response. So CPAs are going to be more, I think, of a membrane issue. However, I could see if those membrane issues farther lead into apoptosis, then it would be helpful. But I think more if you're thinking more along the times of like osmotic shock or changes in osmotic pressure, CPA or the SEPT is probably not going to be super helpful. I hope that answers your question.
Thank you.
Abhilash, go for it.
Yeah. So how do we know that there is a collision happening between the cell carrying droplets and LN2 droplets? Because we are injecting the cell carrying droplets into the stream of the LN2 droplets, the LN2 aerosol. So it forms like a conical stream. So we know that it's into the phase of the LN2 droplets. So that's how we know that the collision is happening between these two droplets, because LN2 droplet number density is high and the droplet is being injected into it. And other reason that we can tell is the collision is happening by this plot, again, the one that I showed here. Here with the pre-cooling one, the blue one, we can see when there is a co-flow present, then the cooling rate is higher. But the gray one, where there is no pre-cooling present, there the cooling rate is lower. So the difference of this is because of the collision happening between the droplets. Oh, sorry. Yeah, this is the plot. So we can see the cooling rate distribution for the two cases, one with the pre-cooling, which is the blue one, and there we have the stream of the LN2 droplets. And then there is a blue one in which we only have the cell droplets. There is no LN2 co-flow happening. So with the gray one, we can see that the cooling rate is lesser than the blue one, meaning that in the blue one, there is extra factor that's coming up and increasing the cooling rate, which is the collision happening. The other part is what are the forces? What forces would bring the two type of droplets together, overcoming the changes, difference in temperature? I didn't understand the second part.
Well, so it is possible that the droplets carry different charges. That's a type of air. And so initially, at least, the difference in temperature is so high that you can have, if you try to bring together a hot droplet with a cold droplet, you can have,
Oh, so
difficulty getting both together. So
in this system, we don't have the charge present on the droplets. They are neutral. And about the droplets being hot and cold, like two different temperatures. So I don't know if that would cause a difference. I I didn't think of it that if the temperature difference would make them move apart.
Well, I think with the charges, you probably
Okay.
think you don't have charges, but there's always when you make an aerosol. That's how the aerosol stays aerosolized. Otherwise, droplets would probably coalesce back into a liquid.
Sorry, say that again. If we have the droplets and
So it is more likely that you have charged droplets than neutral ones. Because that's how the aerosol stays in an aerosolized form. Otherwise, droplets would coalesce and
yeah, so I don't think so that we have a charges here while we are aerosolizing because we are just passing them through our orifice and then vibrating it. So there is no mechanism. Maybe there's no mechanism present that can induce charges here.
okay.
But I can think of it and I can send you a note about it. Yeah.
That would be great. Thank you.
Yeah. Thank you.
All right, awesome. Thank you all for all the lovely questions. I just want to thank you all for coming to our public webinar series. If you aren't already, make sure to follow us on LinkedIn, Instagram, Facebook, and YouTube. And be on the lookout for emails and posts about our next webinar series happening on May 5th, where Dr. Matthew Powell Palm from Texas A&M University will be presenting. Have a good rest of your afternoon.